I'll try to keep it short and sweet. There are many different types of retinal ganglion cells. At least 18 different RGCs have been discovered. They come in larger and smaller ones. The midget RGCs are more dense near the fovea for sharp perception, and the parasol and larger RGCs are more dense in the periphery. Interestingly, rather than just conveying information like other ganglion cells, unique to the retina, they have receptive fields of their own, and they detect changes in stimulation of these receptive fields and increase or decrease signals sent down the axons of a single RGC. Within the ganglion cell layer, there are yet more layers. Similar to the columnar processing in the cortex, layers of ganglion cells process various attributes of images like color, motion, texture, spatial cues, and absolute light. Evidence suggests that these individual attributes are delivered to specific regions of the visual cortex. Rods and cones receive lots of information. By the time the visual information leaves the ganglion, that information has been truncated heavily. It is only being conveyed to the rest of our brain by 1.2 million ganglion cells. When that image was first perceived or received, it was received on 120 million rods and 106 million cone receptors. You do the math. They essentially decide what information is kept and what is discarded. Now, to the question at hand. How does light exposure or lack thereof affect our circadian clocks? This is done with a specialized group of RGCs known as intrinsically photosensitive retinal ganglion cells. IPRGC is the acronym for them. It's quite a mouthful. These IPRGCs are unique because they contain a photopigment called melanopsin. Sorry about that. The pigment is particularly sensitive to blue light. These cells convey information to the suprachiasmatic nucleus, or SCN, of the hypothalamus. As you undoubtedly remember, the hypothalamus controls the pituitary gland, which is the heart and controller of the endocrine system that governs every part of our organism. If the SCN receives information and tries to synchronize with the external light-dark cycle, when the efforts at synchronization go awry, disruption of our sleep will occur. If the disruption becomes chronic, a whole host of dysfunction is sure to follow. Very common would be feelings of depression. So, in addition to doing a bunch of research on the retinal ganglion cell, I also did a bit of research into light therapy and the use of SAD lights, seasonal affective disorder lights. And this is a subject that's very near and dear to me because I am someone who suffers in the wintertime from long, gray, rainy days. And there is definitely consensus here, as you can see, on papers supporting the beneficial effects that one can get from using light therapy. There were many more sources that I drew upon to put this presentation together, but here are but a few. Thank you.